The aim for agriculture, and specifically crop production, is maximum uniformity along with maximum yield. In this lab, we examine the effect of increasing plant density and also the effect nitrogen fertilizer has on the yield. Research in the use and effectiveness of nitrogen is becoming a very important area of study. With over-application resulting in leaching, it is important to understand the best amount to apply to fields to reduce environmental damage and resource overuse, while maximizing yield and ultimately profits. By testing nitrogen applied and seed spacing, it is possible to calculate the best combination of seed spacing and nitrogen application in order to reduce input cost and increase profit margins. With this knowledge, producers will know the most efficient uses of nitrogen and seeds. Therefore, nothing is used in excess and wasted. This is crucial to the environment because the overuse of nitrogen is beginning to leach into groundwater and the atmosphere. Therefore, our hypothesis is increase in nitrogen and seed density will increase shoot mass until maximum yield is achieved, at which point it will continue at a constant rate. For this lab, materials needed would be 16 containers, potting media containing a vermiculite mix, 0-10-10 nitrogen fertilizer, ammonium nitrate, labels for containers, paper bags to collect samples, and shears. Procedures were as follows. 16 different planting plots were filled with sand vermiculite mix, separating the plots into groups of 4. 1 gram of 0-10-10 nitrogen was applied to each plot. Ammonium nitrate was applied to each trays as follows: 0 grams to 1 pot, 0.25 grams to 1 pot, 0.5 grams to 1 pot, 1 gram to 1 pot. Once the sand vermiculite mix is thoroughly mixed in with the ammonium nitrate, we topped off the soil and compacted each plot. Next, we prepared our seed. We needed quantities of 200, 100, 50, and 25 seeds for each tray. You will need 4 sets of these quantities. Apply the seed densities to the 4 pots in each of the 4 trays, labeling seed plots as you go. After 4 weeks in a controlled greenhouse environment, we observed and recorded the health and size of the seedlings in the pots. We clipped the plant's shoot and placed them in a paper bag. Bags were labeled by the sample's seeding density and fertilization treatment. We then weighed and recorded masses of shoots only.
Table 1, as seen here, displays the data our group collected. In this experiment, our independent variables were nitrogen applied, as seen in the first column on the left of the table, and seed density, which is identified in the second column. The shoot mass seen in the column to the right was the dependent variable. This graph, derived from the previous table, Table 1, displays the mass change due to an increase in quantity of seeds and compares the 4 different varied amounts of nitrogen applied, with each color representing a different quantity. The application of 0 grams of nitrogen to the 4 quantities of seed densities show only a slight increase in shoot mass. In addition, the plant showed signs of nutrient deficiencies in regards to their color and size. The fertilizer application of 0.25 grams of nitrogen shows an increase in shoot density until the data shows a slight decrease between 50 and 100 seeds. However, it ultimately increased to continue the trend of increasing mass as seed density and nitrogen are also increased. As indicated by the green line, the fertilizer application of 0.5 grams displays a constant increase of mass as seed density increases. Pots with 1 gram of fertilizer harvested higher mass in every density compared to those with lower quantities of nitrogen. These 4 pots also displayed the greenest color and healthier plants when compared to the other flats of radishes. Table 2 shows the ANOVA test, or the analysis of variance, which is a means of comparison which takes the average mass of all tests and finds a significant difference based on treatments. The p-value is the probability that results can be attributed to chance and not to the treatment or independent variables. Values range from 0 to 1, and with 0 being the best. If the p-value is less than or equal to alpha, then we can accept our hypothesis because it is statistically significant. In this case, we used an alpha of 0.05. Therefore, all three variables, nitrogen application, seed density, and the interaction between the two, are all statistically significant. Table 3 displays the Tukey HSD, a pairwise comparison test of mass for nitrogen and seed variables. This test separates homogeneous groups by applying letters to indicate significant differences. If lines share one or more letters, then there is no significant difference, while if the lines display different letters, they are significantly different. For instance, we see that there is a significant difference between the test with 1 gram of nitrogen and 200 seeds applied compared to all other tests. Our hypothesis was that as we increased nitrogen and seed densities, shoot mass would increase until maximum yield was achieved, where it would continue at a constant rate if additional amounts of the variable were added. We accept our hypothesis, but with reservations. Due to the findings that every variable was statistically significant and had a significant effect on the outcome, or mass harvested, we accept our hypothesis that as we add nitrogen and seed density, the mass increases. However, we also want to note that our study did not display a maximum yield and an evening out of mass at a constant rate, since in general all tests increased. Additionally, our study occurred in an environmentally controlled greenhouse, with only one variety of plant. Further research could occur in the field in order to account for environmental differences and a diverse plant variety. Ultimately, this study identifies the importance nitrogen and seed density has on plant growth. Producers can use this general knowledge and mimic this study in order to find the best production and input levels to maximize profits and reduce input costs, while protecting the environment from leaching of nitrogen due to excessive application.